This is a lab activity you're going to conduct at home this weekend, and very easy to do, very simple materials. And the main idea is that you're going to just go through these procedures, and you're going to record observations. So you're going to see changes that occur, and you can do this in any number of ways. So let me get started and I'll show you. So this is on osmosis and diffusion. So when you do this activity, you can take pictures, and submit those. You can do a short video. If you like doing a little PowerPoint where you can put your pictures in, that's great, just to show what happens. So your materials are very simple. You need 2 glasses or containers. They can be little mason jars. They can be water glasses. It doesn't matter. But in those glasses, you're going to put something. So you want to make them at least big enough that you can put whatever it is you've decided to put in there. One of the glasses you're going to fill with just tap water, or even better, distilled water. And then you're going to label that in whatever way you want. So you can just put it onto a piece of paper and write right below it, this is your hypotonic solution. Okay? Good. Next one, you're going to do the same thing. You put water in the glass, but using warm or even hot water is going to help you to make a solution that can dissolve a lot of solute. The solute you're going to put in there is either sugar or salt. Just put in as much as you possibly can, keep stirring, and get that to dissolve until you have what's called a hypertonic solution. Good. Now, you have a choice. You want to use either fresh vegetables like potatoes or carrots or even celery, or you can use eggs for this next part. So if you take a vegetable, you want to use a fresh one. You don't want to take it out of the freezer, but take something that you got in the grocery store. Potatoes work really well, but I just happen to have a carrot here. And you want to create some slices of it that are fairly equal in size and width. So I'll just show you because I did that here already. So here I have a slice, if I can do it like this. Here's a slice of this carrot. I tried to make it as even as I could, and I made a number of these same size so I could use those in my experiment. The other option are the eggs. So this is a raw egg, and this raw egg I removed the shell by putting the raw egg into submerging it into vinegar. So I took a cup, put the egg with the eggshell in it, poured vinegar over it. White vinegar works really well, and left it for at least a full day, if not a little longer, and then washed the shell and removed the shell, which you'll see all sorts of bubbles on the shell, and ultimately the shell erodes off. And then I took it out and kind of gave my egg a little bath and took all the rest of the shell that was stuck on there still off. You have to be careful. And then you can see, I don't know if you can see it, but I can see that egg yolk inside, and it's still very raw. So it is now my model for a single cell with the membrane, and the inside is the cytoplasm, and you can kind of say that the yolk is sort of representing the nucleus, although it's not truly that, but this is a perfect thing you can use. So if you do this, you want to do at least 2, maybe 3, just in case one breaks. And then once you've rinsed it, set it aside. But do this, if you like the egg idea, do it right away. Get a couple of raw eggs, put them in vinegar, and let them sit so you have time to get the shells to come off. Okay, so now you've got that. Once you've done either your eggs or your vegetables, make your observations. So what you want is a pre-test and a post-test. So you can take pictures, you can weigh it on a scale, you can take a string and measure the diameter of your egg, you can put it next to a ruler, you can do anything you want that actually gives some indication as to what it looked like before you did anything else to it. So its texture, is it flexible, etc., etc. Then you're going to put the vegetable slices or the eggs into each of those solutions and just put it all the way in so it's fully submerged. And then wait 24 hours or so, at least 24, but it's variable. Then take those out of those solutions one at a time. And as you take them out, remeasure, do all the measurements once over again, and do the length and the width and the, if you have a kitchen scale, put it on that, see if you can measure the weight, that's really nice. Or do something that you can show that process, that what happened, where your before is. Hopefully you took pictures, you did that on a video, and now you're looking at the after. Now, what you're doing is looking at some of these concepts that are in OpenStack. So we're looking at this idea of tonicity, which is basically what happens, how this extracellular solution that you put your egg or vegetables in will change the volume of the cells in the vegetables or in the egg by affecting where water flows. That's osmosis. When we use the term osmolarity, so that's tonicity. When we use the term osmolarity, what we're doing really is what you did in OpenStax, which is measuring what the total number of particles are in a solution versus the number, the actual water in that solution. So a solution that's going to have a low osmolarity means that it has a lot of water compared to things dissolved in it. Something that has high osmolarity has fewer water molecules as compared to what's the solute is, what's dissolved in it. So the idea of tonicity, when we talk about that, the volume of the cells relative to, this whatever solution you're putting into, is going to obviously be very related to the idea of osmolarity. So have fun with it, and here's one other picture that might be helpful, just to look at these. These are red blood cells on the left that are being placed into three different solutions, one that's hypertonic, one that's isotonic, one that's hypotonic. And then same thing on the right, except for now we're looking at plant cells. So you can kind of see how that works. And have fun, and we'll discuss on Monday.